In these videos, we are looking at cell components. This central vacuole is central to the way that plants function, and I think it's often overlooked by or undervalued by people when we're looking at plants and studying plant biology. So the central vacuole serves many important functions. Often it just gets written off as being part of storage, but a huge role of the central vacuole is in structural support. So look at this plant cell here. See how most of it is composed of the central vacuole? If it were just for storage, it probably wouldn't take up so much of the cell, depending on the plant cell. But most plant cells have this huge central vacuole. You can think of a plant cell as a cardboard box with like a paper bag, not a paper bag, a plastic bag inside of it. So the cardboard box is the cell wall. It's somewhat rigid. Then you have this plastic bag inside of it. That is the plasma membrane. And the plastic bag has no structural support. So to give more structural support to the plant overall and to that cell in particular, we can basically blow up a balloon inside that plastic bag. And that is the central vacuole. That balloon that pushes the plastic bag out to the walls and provides more support for that cardboard box. Because if you were to try to push on a cardboard box, it would be pretty easy to collapse. But if you put a blown up balloon inside there with pressure, then it's got a lot more support, a lot more rigidity. So plants that have a completely swollen central vacuole are in what's called a hypotonic condition. And for plants, that's perfectly normal. That's where they want to be. It provides their cells this structural support. When plants are in a condition where it's hypertonic, where there's more solutes outside of their cells than inside of their cells, that causes water to be drawn out of the central vacuole. And you can see that when water is drawn out of the central vacuole, the plasma membrane collapses inward. That's plasmolysis or plasmolysis, and it causes this collapse of the cells. And when you see plants that are wilted, they are in this plasmolyzed condition. So that could be because there is a lot of solutes in the soil, so a lot of salts that are pulling water from the plant by osmosis, or it could be because the soil is incredibly dry, and that could cause water to be removed from the plant just from diffusion. So this pull of water, and I suppose it could also be considered osmosis too, but this pull of water from a place of high water inside the plant, plants are normally 90% water, to a place of low water. So when plants wilt, they're in this plasmolyzed condition, which could be caused by a hypertonic soil or from just a lack of water in general. So plant cells in their natural condition are hyperswollen, full of water, and that's what gives them this structural support. And that hyperswollen condition comes from the central vacuole. So having a central vacuole is essential to the plant's overall structure and being able to support itself. It also functions for storage. So plants that have toxins might store them in the central vacuole. They're trying to prevent herbivory. There's also a pigment that some plants have, or many plants have, that gets stored in the central vacuole called anthocyanin. This is the purple pigment, but it can also be, anthocyanins change color based on pH, which we'll do in one of our labs. But they can go from blue to green to pink to yellow, but purple is sort of their base neutral condition. And those are stored inside the central vacuole. So here, our central vacuole, we can see in the swollen normal condition and the plasmolyzed condition. So we'll look at this in our lab about water and roots, and we'll look at this with Elodea cells. We will have them in regular water, and they'll look like this, fully swollen. And then we will have them in a saltwater condition, and they'll look like this. So this would be a turgid plant cell, and this would be a plasmolyzed plant cell, where the plasma membrane collapses inward and pulls away from the cell wall. And that happens because the central vacuole shrinks, because water is lost from there. So the membrane surrounding the central vacuole is called the tonoplast. So if we were to look at sort of layers within the cell, we have the cell wall on the outside. Just inside that we have the plasma membrane, which is kind of represented in this brownish color here. Then we have all this cytoplasm. Within the cytoplasm is our central vacuole, which is surrounded by the tonoplast. So we have cell wall, plasma membrane, tonoplast. Let's look at cell walls. All plants have all plant cells, have a primary cell wall. And I say this with a little asterisk because I'm always worried about saying all. Perhaps there are some plant cells that don't have a primary wall. This also refers to how all plant cells start. All plant cells start with a primary wall, but it's possible over the course of that plant cell's maturation that's going to lose the primary wall. So all plant cells start out with this primary cell wall. And that primary cell wall contains cellulose, as well as other compounds that give it some rigidity as well as flexible support. So hemicellulose is very similar to cellulose. These are present in these fibrils that are crisscrossed throughout the cell wall. And then pectins are these highly branched polysaccharides. So it's like if starch were super branched, and that makes this more gelatinous, flexible structure. And that sort of surrounds the cellulose and hemicellulose kind of skeleton. So if you think about this as like a building compound, this might be like rebar and a more concrete type structure. Overall, giving it some flexibility and some rigidity. You can see there's a middle lamella here that represents this pectin-rich layer that helps stick adjacent plant cells together. So when we look at multiple plant cells, they're going to have some pectin in between them called the middle lamella. And these are often sort of sheets, which is why they're called lamella, like laminated. So all plant cells can start out with a primary wall, but later in some evolutionary groups, we're going to get plant cells that develop this secondary wall. And the only plant group that doesn't have this would be the bryophytes, because we start to see this secondary wall when we start to see lignified vascular tissue. So the secondary wall forms within the primary wall. So here's our original primary wall here. And the secondary wall has a similar composition, but now we are exchanging this sort of pectin flexible for this rigid lignin. So we have much thicker wall, and it has this structurally rigid lignin in it. And that lignin is super hard to break down. So now we have this really cool cell that has this like woody wall to it instead of this kind of more flexible cardboard-like primary cell wall. And this is composed of multiple layers, which you do not need to know those multiple layers. So when a plant cell forms a secondary cell wall that is lignified, that plant cell eventually is going to die. So the primary cell wall helps deposit this secondary wall, but as it gets deposited, it cuts off the internal, like plasma membrane, cytoplasm, everything within that cell is cut off from the exterior environment. And what's eventually going to happen to that interior is it's going to die. These cells are not photosynthesizing, they're not making their own energy, and now they don't have input from other cells to help keep them alive. So these become empty tubes. We get this rigid, woody secondary wall, and then an empty inside. And what that can be used for is a variety of purposes. We saw it in cork, which forms this external barrier around some plants called bark, and that bark can help reduce impacts from herbivory because it's dead, right? If you surround yourself in a bunch of dead material, like an armor, it's not really going to matter if it gets eaten because those cells are dead. There's nothing really in there for some herbivore or some herbivorous organism to eat. So it's a barrier in the sense that you're not damaged from herbivory. And it's also a barrier in the sense that you can protect yourself from other external things like potentially frost damage or fire damage. And those cork oaks grow in an area where there are periodic fires, a Mediterranean climate like we have here in California. So that's why they have such thick, corky outsides, is to protect themselves from fires. Other cells that we might see that secondary wall in are cells that are going to be transporting water. So the water is only going to move in one direction, from the roots up to the shoots and then out the leaves. So we don't need any control on how that water moves around. So the cells that move it can be dead. So essentially they're like water pipes. They have a rigid exterior wall and empty insides, and that allows water to be moved through them with great efficiency because there aren't any membranes that it has to be pushed through. That reduces the amount of filtration that happens of that water, but it increases the speed that that water can move around. Here are some images of those cells. So the stains of our prepared slides are different than the stains that we'll use at home. The stains in the prepared slides of the images I show are stained red when they have a secondary wall. So these are vessel elements in the xylem. They're water transportation cells, and they have a thick secondary wall. And with this particular stain combination, the secondary wall turns red, and the primary wall turns blue, or blue-green. So all of these cells out here are just regular cells with a primary wall. It's these cells in this X in the middle that have that secondary wall. We'll look at why these ones look a little bit different when we look at roots. Here we have another bundle of cells with really thick secondary walls. So in the slides that I show you in lectures, often these secondary walls will be stained red, but in your stains at home that you're going to be making, you're going to use toluidine blue, which I always have trouble saying, and that stains the primary wall like a dark purple, and the secondary wall is going to be this like aqua blue color. It's really beautiful, but you'll have to rinse your slides pretty well in your samples to remove more of that excess dye so that you can see the stains just in where they have picked up to and not just in the exterior water. So these cells would also have that thickened secondary wall. All right, I think I'm just going to try to blast through this last part. So this is a cell-to-cell interface. We've looked at individual plant cells, but how do they interact with each other? Because plant cells are going to be produced in place, and then they're not going to move around the plant. They're going to stay where they are. So to communicate with each other and to sort of create this functional multicellular organism, plant cells get stuck together, and they get stuck together with this material called the middle lamella, or middle lamellae, which is rich in pectin, which is this sort of gelatinous, flexible material. And it glues the adjacent cells together. So that gives the plant even more structural support, because now we have all these cardboard boxes with inflated balloons inside that we've then glued together. Within that whole structure, so we have one cell wall up here, another cell wall down here. We have the middle lamellae gluing them together. But then we need those plant cells to be able to interact with each other. So to do that, there are channels through the cell walls and through the middle lamellae. And plasma membrane would traverse through these channels and connect the cytoplasm of the two adjacent cells. So materials could be moved through these, cells could communicate with each other through these channels. And these channels are called plasmodesmata. Plasmodesma would be a singular channel. So this is a plasmodesma. And here we have many plasmodesmata. So plasmodesmata, it's its own plural, I don't have to put an S on it, are channels of cytoplasm that connect adjacent cells. And you can see this with this micrograph of cells here. We have the contents of where the cell would be. These are empty though. We have these really thick secondary walls. Oops, I moved it. And then those secondary walls have channels running through them. So these are the plasmodesmata. If these cells are dead, the plasmodesmata become pits. So they become empty tunnels that still connect these cells. And we'll see this when we look at the sclerids in pairs. you'll see these stripes in that secondary wall. Those are pits where Plasmodesmata used to traverse and connect that cell to the cells around it. But that cell eventually dies, and so its plasma membrane goes away. It no longer has this living cytoplasmic connection to the cells around it. But that's part of how these dead cells continue to function and form, is because they can be fed from cells from the outside through a plasmodesma. So plant cells have cell walls. Unlike the plasma membrane, this rigid structure does not allow the flow of materials. Plasmodesmata are tubes of cytoplasm that connect adjacent cells. These tubes travel through pits in the cell wall, and these pits can be simple, just as a tunnel with kind of the same diameter all the way around. Or, in some things, you'll see a bordered pit. So bordered pits, I guess I usually draw this, but I don't have drawing capabilities right now. So bordered pits sort of have these swollen entrances where the entrance is kind of skinnier than the actual pit itself. And that just helps us for identifying types of cells. When you look at a bordered pit kind of straight on in a really good microscope slide, you're going to see this donut, the donut that sort of surrounds the pit. But if you're just looking at a simple pit, then it's just a hole, and it doesn't look like anything special. Okay, so it's unlikely you'd have that level of detail in your homemade microscope slides, but potentially you will. And that is the end of cell components.